it always takes me back. It always takes me back to, when I was in Ghana, when I got the invitation, it gave me a very strong memory that I'm going to pour back what I've learned over the past 4 years to, people back home. And I'm very much excited. Yeah. So thank you, everyone. And to the participant, I'm very excited to have you all dedicate your time to, hear what we have here ongoing. So, without wasting much time, oh, well, thank you for the great introduction about my profile. And I don't think I need to mention anything because he's already said more than, enough. So I'll just go ahead and, go to the presentation. Today, I'll talk to you about cancer immunology in the first half and then hit deep into the cancer neuroscience, which is something that a field in cancer which emerged recently that amazes me so much. And today I'll talk to you about more mechanistic insight. And basically, when I say mechanistic insight, studies and data that has been published recently that are very interesting, which I would want to share with you. And some clinical translation and ideas, most people for like you guys in Africa can, adopt and investigate if you are interested in this field. So my talk is going to be divided into two parts. First part is going to talk about cancer immunology and some immunotherapies. It's going to be boring for the first 10 minutes because cancer immunology always seems to be very broad and it's hard to, present anything in cancer immunology. So it's going to be very brief, but it's not going to be as exciting as the second part, which is cancer neuroscience, which I will take my time to delve you into the emergence of cancer neuroscience itself, what it is, and why this field is so important that people are investing their time, money, and a lot of resources into it. I would like to put these disclosures and declare my interest. I have no conflict of interest to declare, but I want to tell you all that, and anybody who have access probably currently or later to this material, that this presentation is intended solely for educational and academic purpose. Some, which I would say most or 100% of the figures, schematics, or images are adapted from previous publications and literature. So I do not have any copy. I do not owe this material. So copyright for this material remain ownership for the original authors and the publishers, and I do not claim ownership. Yeah. So Oh, one thing I would also like to, what do you call it? Mention and add to this point is that none of this data or experiment I'm going to show here is anything I ever did by myself. No. Nothing, and my whole project has not been published yet. So unfortunately, I'm not able to share any data from my own and personal research. So anything I'm presenting here today is exciting research which has been going in the field of cancer immunology and cancer neuroscience, and I do not own any ownership of any of this data set. So let's get the whole thing going. And this is what I'll do. Just feel free to write your questions. And at the end, I'll be more than happy to ask your questions. So if you have any question, write it down and I'll be happy to take it at the end. This is going to be a very short, but I hope it's exciting, at the end of it all. So I'll start with cancer and the hallmarks. So, since I'm going to talk about cancer immunology, cancer neuroscience, I want to start with the heart of the disease, which is cancer. Orwell started by already introducing the statistics of cancer. So, why we study cancer? Because it's very common. The prevalence is so high. I didn't put any prevalence data here because I'm not an epidemiologist, so I wouldn't want to waste too much time. But most of us know that in Ghana, if we are taking ourselves back to the last 50, 100 years, there was few cancer cases, and the cases has been on the rise for the last recent years. So that is why it is very important for people like us, and including you, to be interested in study cancer so that we can find cure for cancer. So what is cancer and tumor? I just put these two things here because people we use them interchangeably, but some people don't even know the difference. So cancer is a large group of diseases characterized by uncontrolled cell growth. Um, while tumor is an abnormal mass. So when you have I'll use my pointer. So here you have a normal cell, and when the cell is defective, there are several things that can happen, which I will not. This is not a cancer biology talk, so I will not spend time going to the deep of the process of cancer formation. And no, that's not. That's a topic for something different. So if you have a normal cell, if that cell is transformed, it become a defective or transformed cells. And what happens to this cell is they begin to multiply huge. The multiplication, they just divide. And that is what we call this whole process. And at the end is what we call cancer. But when the cancer happen, they can form, when your cells continue to proliferate and grow, divide over time, they form a very big mass of tissue. So and that is what we call tumor. So for instance, a normal breast, I'll use breast as example because I work on breast cancer, but you have normal breast cells when they are exposed to radiation, DNA damage, anything, a lot of things. I don't want to go into causes of cancer, but upon exposure, that can lead to DNA damage. The cells can be transformed, become cancerous. So what happens is they divide. And after they divide, they can lead to abnormal big or mass of tissue. That is what we call tumor. So basically, that's the difference. And cancer is characterized by when there is cancer in a specific tissue or an organism, human being, what happens is there are hallmarks or characteristics of that cancer. There are about 14 of them, which we will not look at them one by one. But today, we are going to specifically focus on one most important, long-lasting hallmark of cancer, which is avoiding immune destruction. So today, that's what we are going to look at. So that takes us, so one of the hallmark which has really, really, really evolved over the last 50 to 100 years is avoiding immune destruction. And just bear in mind that this hallmark of cancer keep changing. It started from very little known hallmarks and it keep expanding. And I'm sure that in the next 20, 30, 50, 100 years, there may be more than 30 hallmarks of cancer due to active research that has led to interesting discoveries. So I'll start with cancer immunology. And what I'll talk about with respect to cancer immunology is I'm sorry, I need to close something real quick. Yeah, so what I'll talk about in cancer immunology is to start telling you what exactly is cancer immunology. So it's basically the study of how the immune system interacts with cancer. Like I already said, the first part, which is cancer immunology, is quite boring because it has been existent for the last 50, 100 years. So there is nothing new. So I'll run through quickly so that we can get into, the cancer neuroscience, which is more exciting. So cancer immunology, basically looking at how cancer interacts with the immune system and how the immune system interacts with cancer. And why should we study cancer immunology? This is because some evidence over several years have established that there is a very clear evidence that the immune system has something to do with cancer. So an example is that people with HIV, we already know that their immune system they are very immunocompromised. Their immune system is not 100% functional. And what happens to these people is that they are exposed to several cancers and viral associated cancers. So this is a clear evidence that if your immune system goes wrong, you are at prone of getting cancer. So that suggests that if your immune system is intact, then you have high chance of fighting cancer. So that is in human example that can back that cancer immunology is indeed real. Another one is preclinical studies in mice, which we most people across the world have shown that mice that lack specific immune cells or any component of some component of the immune system, they die faster or they even develop more cancer. And even if they develop cancer, they die faster compared to mice with intact immune system. An example here is RAC2 knockout mice. The RAC2 knockout mice, these are mice that do not have T cells and B cells. TCR knockout mice, these are mice that do not have T cells. Athymic mice, these are mice that do not have thymus. And if you know a little bit about the immune system, which I didn't start because this is not a basic immunology lecture, but athymic mice do not have adaptive immune system, which is especially T cells, because T cells are produced in the bone marrow and they mature in the thymus. So if you take out the thymus of a mouse, the mouse become athymic and they do not have any T cells. What happened to these mice is that they develop cancer faster, they die faster. This evidence, together with what has happened in humans, have made us believe that the immune system indeed is important for controlling cancer. So, before these studies, in addition to this well-known clinical association and the knockout studies in mice, there is this immunosurveillance theory which says a physiological function of the immune system is to scan, because we have already, so these first few 3 points have established that the immune system indeed fight cancer, because in the absence of the intact immune system, people mostly get cancer, or you even die faster in cancer. So, therefore, what does the immune system do? That leads to the immunosurveillance theory, somewhere what was proposed in the 1950s, and it says the physiologic function of the immune system is to scan, recognize, and destroy cancer cells, clones of transformed cells. Remember, I already said a cell must be first transformed till it eventually evade the cell division, then it become cancerous. So the function of the immune system is to scan, recognize, and destroy this clone of transformed cells before they, grow into tumors. So basically, an overview of the immune system, telling you that the immune system is indeed important due to this evidence available. Now, I have a very big question, is that if the immune system is important and can surveillance and kill tumor transform cells before they even become cancerous, how does the immune system distinguish cancer cells from normal cells whilst it's scanning or killing? So and that leads us to what we call tumor antigens. So without recognizable tumor antigens, the immune system cannot distinguish between a normal cell and an abnormal cell. So what I'm trying to point out here is that before the immune system can kill or find the cancer cell, there must be antigens from the cancer. So there are two types of antigen mainly studied in cancer. Tumor-specific antigens, they are mostly found only on tumor cells and not healthy tissues. Then we have tumor-associated antigens that are shared between, both. You can find them both on normal tissues and abnormal tissues. So how do we get these tumor antigens? Because remember, I already said without antigens or tumor antigens, the immune system is useless. So how do we get them? So one, the main thing people do is if you treat with chemical, which is drugs, chemotherapy, if you use radiations or targeted therapy, these are all drugs that are used to treat cancer. And the goal is to kill the cancer cells. But the problem with this treatment is that if you kill the cancer cell, they still come back. So what happens is when you use this treatment options, you actually you break the tumor cells down, then you release antigens. Then these antigens can actually be picked up by the immune system for, immune response. So what happens is we can also enhance antigen presentation, because when we have the antigens now I've already said ways to get the antigen is through this kind of therapies. But if we get the antigen, what next? We can enhance presentation through cancer vaccines and stuff that can activate the innate immune system. So basically, a summary of this is that without antigens, the immune system is useless. So one angle of research is how do we find antigens? So if you are here and you are interested in pure cancer immunology research, you should start be thinking about how we can get antigens to be presented to the immune system so that immune system can become functional. So that break. Somebody wants to talk? Can this person probably be muted? All right, thank you. So this brings me to now we have the antigens because I said you have the cancer cell or the tumors, which is a huge mass sitting somewhere in your body, in your breast, your lungs, anywhere. How do you get antigens? How does the immune system get access to them? I said first, we need tumor antigens. When we get the tumor antigens, what next? We need immune cells to recognize and then process, kill them. So cells of the immune system, that brings us, it's the heart of the immune system. That is what I'm going to tell you. Without the cells, you are useless. So because why I'm saying this is because the immune system is made up of the organs like the spleen, the lymph node, the skin, Peyer's patches, a lot of things. But what I have here is that um, the heart of the immune system is basically the cells that moves from one place to another to recognize the antigens I've already spoken about in this previous slide. So the cells of the immune system, this is an oversimplification, but we have the cells that belong to the innate immune system and the cells that belong to the adaptive immune system. There are some cells which sit in between because they can perform some functions related to both the innate and adaptive immune system. But we're going to talk a bit more about them later. But this is an oversimplification. So let's get into the deep part of it. So the cancer immunity cycle. So now I already said, just to summarize, the whole, how does, the immune system recognize cancer and kill them is that first, this is the big tumor sitting here. We already said tumor is abnormal mass. And if you have the tumor sitting somewhere here, I'm sorry, I need to move this. 1 second. So if you have this abnormal tumor big thing sitting here, the immune system is literally useless. It cannot do anything. So what you need to do is, first, we need to release cancer antigens. So we need first the antigens. And some of the cells must die. You must kill some of the cells so that when they die, they break down, they release their antigens. And I already mentioned that for us to be able to do this, we need radiotherapy, chemotherapy, or targeted therapy. So with these therapies, and mostly they are the first line of treatment before immunotherapy. So what happened is so if you do this, first you must release the tumor antigens. If you release the tumor antigen, the second step is we need the antigen to be recognized by the killers of the immune system. And the killers of the immune system are the T cells. So the innate immune cells, like antigen presenting cells, example is dendritic cells, will then pick this tumor antigen. So the tumor cell will be, first it must be broken down, release antigens. The antigen is being picked up by the antigen presenting cell, and I'm saying example is dendritic cell or macrophage. And what happened is they travel to the secondary lymphoid organs where the T cells sit. They are plenty, they sit there. That's the powerhouse of them. And then when they get there, the antigen presenting cell give these antigens to the T cells. When T cells are there, they don't know anything. We call them naive. Then upon receiving the antigen, they become activated. So we call them activated T cell. As soon as they are activated, they realize that something is wrong with, let's say, let's assume, I'll take it again. Let's assume you have breast cancer. There is huge breast, tumor inside your breast. What happened is first, without antigens, the immune system is useless. It's naive. It doesn't know anything. So first, you must release the cancer antigens. Upon release, what happened is they are picked, the antigens, which is these small ones, must be picked. They are picked by antigen presenting cells. Example is dendritic cells and macrophages. When they pick them, they cannot kill directly. They take them directly to the killers, the professional killers. And I say they are the T cells. And what happened in the T cell, they do not directly sit in the cancer. They actually sit in the spleen and lymph node. And they are naive. Naive means they don't know anything. Just take a naive human being. He doesn't know anything. And upon receiving the antigen from the dendritic cell, then they become activated. They realize that something is wrong in the breast. Then they just enter the blood vessels. Then they traffic back there. So they call it trafficking. Then upon when they get, so step four, first get the antigen, dendritic cell will pick the antigen, traffic to the lymph node where the T cells are, present it to the T cells. They traffic back to the tumor, and when they get to the tumor, they recognize the tumor and they kill the tumor. So that's the cancer immune cycle. And upon killing the tumor that will die will further release antigens. Those antigens are being picked again by dendritic cells, taken to the lymph node, presented to the T cells, trafficked back, and the cycle continues. But trust me, if this is what happens, and our immune system is intact, then nobody should die from cancer. So when I go into details, you realize that at every step, the cancer cells are very wild. They find ways and means to break this, break this, break this. They can break any of the steps you see so that the end results of the T cells coming to kill them won't work. And I'll talk a little bit about that today. So I'm taking an example. So this one was a general cancer immunity cycle, but I'm taking what does an immune response look like in cancer? So the immune cells, like I already said in this slide, there are several immune cells. Each of them have a very specific way in which they kill or they tackle or they handle cancer. So in the cancer immunity cycle, different cells have different roles. They do stuff specifically and they are very different. But I'm going to so for instance, in the innate immune system, dendritic cells, macrophages, NK cells, innate like lymphocyte, they all do something different. So I'm going to tell you about CD8 T cell as a classical example, but just pay attention that each cell does it different. Then adaptive immune system, we have CD4 T cells, CD8 T cells, and B cells. Each of them also have a unique way of tackling or interacting with the tumor cells. So I didn't have time to talk to you too much about each of them because I could tell you about how dendritic cells, each role, macrophage, NK cells, CD4, CD8, B cells. I don't have time to talk to you about each of them individually, but I'm taking a CD8 T cell as a classical example. And why I chose a CD8 T cell is that in cancer, especially, specifically, CD8 T cell, for instance, in an infection like COVID-19 infection, B cells and antibodies are very important. However, in cancer, one of the most interesting cell that is very critical for killing cancer is CD8 T cells, and that's why I pick it to demonstrate the cancer immunity cycle here for you. So first, you have a tumor cell here. And like I said, the first step is the tumor cell, immune system cannot just see a tumor cell. No. It must first, it will definitely recognize antigens from the tumor cells, especially CD8 T cells. They can only recognize antigens, which are in the form of peptides or proteins after it being broken down. So upon the antigen release, the dendritic cell pick up the antigen, present it to a naive CD8 T cell. So this is a T cell, this is a dendritic cell. So this part, I said there is antigen presentation, and I'm zooming in to show you what happened. So you have a dendritic cell and a naive T cell. Naive T cell is a T cell that has not seen antigen before. It is happy sitting in the spleen or the lymph node and doesn't know what is happening in your breast or in the lung cancer or in the renal cancer or in the brain tumor. So what happened here is that the dendritic cell pick the antigen, travel, and give the antigen, present the antigen to the naive T cell. Upon getting the antigen, the T cell become activated, it proliferate, and it migrate to where the tumor is. So CD8 T cell, for instance, when it gets there, it produces two chemicals. Perforin, it can initiate killing by multiple ways, but one of the way CD8 T cell is to produce perforin, which will create holes on the tumor cell surface, and then granzyme B, which can enter the holes and then kill the tumor cell. Another way is to produce effector cytokines like TNF alpha interferon gamma that can kill, cancer, the cancer cell. So this is a typical example of a CD8 immune response in cancer. Um, I don't have time to talk to you about CD4 T cell response, but it's not different from this. It's similar. But B cell are very tricky. They produce antibodies which do not directly kill cancer. There is not too much about B cells in cancer, unlike infection. So I didn't have time to talk to you individually about this, but take the CD8 T cell as example. Now I have a big question for you all. What happens to this CD8 T cell after killing the tumor? Should should it continue on killing, or what happens to it? What happened is my answer for you is that if you don't stop it, it's going to continue on killing the cancer cell, and when it's done, it's going to start killing your normal tissue. So let's say a CD8 T cell upon activated gets to the breast cancer. Let's say it's done killing the cancer cells. It's still active. If you don't break it, it's going to continue on killing. So what happened is the immune system has a way of stopping this whole thing when it's necessary, and I'll talk to you about that later. So we are now already talking to you about cancer immunity cycle and CD8 T cell response. But still, if this is working, why do we die from cancer? Cancer is very stubborn, which I already mentioned. There is an immune invasion in cancer. Cancer has mechanism in which it used to invade the immune system. When I say invade, like it escapes the immune system. And I'm giving you two typical examples is T cell exhaustion and immunosuppression. Just based on the last slide I spoke about, I said when there is an activated T cell, they can go to the tumors, and what they do is they start killing the tumor cells. And what happens is, after killing, what happens to this cancer cell? They upregulate molecules such as, we call them checkpoint molecules, okay? An example is PD-1, TIM3, LAG3. A common example is PD-1. And what happened is, before cancer, what happened is, when T cell is activated, it expresses this PD-1. And the reason why it expresses that, so that when it is done killing, we can bring what we call PDL1. So PDL1, PD1, program def1, PDL1. And as soon as PDL1 binds to PD1, the cell stops killing. This is what our body used to break, at least tell the T cell that please don't kill too much, else it will start killing your normal tissues. But guess what? Cancer cells are very stubborn. They can produce PDL1. So what happened is, now this your CD8 T cell is activated, coming to kill. And what happened is the tumor cell will just produce PDL1, which will bind to the PD1 on the T cell, and that T cell can become what we call exhausted. It just feels like I'm tired. And what happens to this after PD-L1? They can just die. Like I said, program def1. So the T cell will start dying, or they become quiescent. They will just stop working. Basically, a simple term. So this is an activated T cell that expresses PD-1. But if the T cell is exhausted, you see that they upregulate several of these checkpoint molecules. Example is PD-1, TIM3, LAG3, CTLA4, and a lot more others. So what happens is, the immune system can produce PDL1 that can bind to PD-1, and the impact is that the T cells become exhausted, especially in cancer. That's one way the immune system evades cancer. That's one mechanism in which cancer evades the immune system. Another one is the immunosuppressive tumor microenvironment. While some of the immune cells are important for killing and important for trafficking, killing tumor cells, there are some other ones, like some macrophages that can inhibit NK cells. NK cells also kill tumors, like what I just showed you. And some macrophages can inhibit the activity of NK cells. We have what we call myeloid-derived suppressor cells, we call MDSCs, and they can also suppress and inhibit the activity of this CD8 T cell that can kill. One more important cell is Treg, which we call regulatory T cells. Naturally, their role is to stop killing by, the CD8 T cells so that they don't destroy our normal organ. And what happened in most cancers, which I work on, my project and my whole PhD is studying, Tregs. They can produce molecules such as suppressive cytokines, such as IL-10, that can inhibit or suppress the CD8 T cell you see here. Tregs can produce IL-10 so that they can no longer function. They suppress them. They can produce CTLA4 that can block the activity of activated or effector CD8 T cells. So what I've told you so far is that the tumor cells, the immune system cannot recognize antigens unless sorry, the immune system cannot recognize tumors unless there is an antigen. And when they recognize antigen, they present it to T cells that can kill. Example is a CD8 T cell. But the immune system, but the tumor or cancers are stubborn. They have mechanism to escape the immune system. And two examples I gave is T cell exhaustion and immunosuppression. So what are the clinical implications of this? So based on this, research have targeted things in all these pathways so that we can get a better immune response, so that patients do not die, people who have cancer do not die faster. So first thing is T cell specific cytokines. First therapy, which I will not talk about today, but there are cytokines like IL-12 that can activate T cells. Another example is checkpoint blockage. I already mentioned that PD-1 is a checkpoint. And when T cells are activated, they activate PD-1, which is a checkpoint. And like I said, tumor cells can produce anti-PD-L1 to inhibit it. So to avoid that, we can actually block the PD-1 on the T cells after they are activated so that they don't get exhausted. So we will talk about checkpoint therapy. Cancer vaccines. What cancer vaccines do is they ensure they enhance antigen presentation. Adoptive transfer of modified T cells, which I didn't talk about, I'm not going to talk about today, but there's a whole booming field about CAR T cell, and I think another speaker will talk about that. Then there are some chemokines that will help the recruitment of T cells to the side of the tumor. And there are bispecific T cells that are engineered to directly go and kill the tumor cells. So first data I'm going to show you is, which I'll spend much time, is in immunotherapy, is immune checkpoint. So this is a very interesting data that has really, really given hope for patients, especially with melanoma. And what happened here is that this is a very big clinical trial published in Lancet, which showed that if you block with anti-PD-1, especially combination with first and second line treatment, the patients survive longer. So this is a survival curve. And whenever we start with equal number of patients, and whenever a patient dies, this curve drops. So whenever a patient dies, the curve drops. And what will happen is the single treatment, the patient dies faster compared to when you have two drugs which inhibit anti-PD-1 in melanoma. So this data, which is published in Lancet, actually shows that blockade with anti-PD-1 increase the survival rate in melanoma. Another example is a combination of anti-PD-1 and CTLA4. You see that the red line is far away from this, and the patient died less faster compared to a single anti-PD-1 therapy. So this data is telling us that checkpoint blockage, just by understanding cancer immunology and the immunotherapies, the people were able to engineer antibodies that can block these checkpoints. And what happened is the patients survive longer compared to when they are not treated, or when they are even treated with a single therapy. So this is a very interesting clinical data I presented to you. But one challenge here is that Immunotherapy doesn't work for every patient. In as much as this data it looks very promising and exciting, and there are a lot of them which I'm not going to talk about today, immunotherapy doesn't work for every cancer patient, and there are a few reasons. Why? Because there are immune desert, and when I say immune desert, there are some immune tumors that have almost no immune cells. So if you've never heard this before, immune desert and cold tumors, immune desert means like you go there are no immune cells. So that is immune desert. And cold tumors is that the tumor is dominated by suppressive cells and not much of effector T cells or immune cells that can kill the tumor. An example of these cold tumors is pancreatic ductal adenocarcinoma or pancreatic cancer and glioblastoma or brain tumor. Another reason why it doesn't work well is low expression of checkpoints. Some tumors do not express. So, for instance, there are also anti-PD-L1, which we use to block, the PD-L1 expression on the tumor cells. And some tumors do not even express PD-L1 at all. So, if you are treating with anti-PD-L1, what are you targeting? Literally nothing. Another reason is that the tumor is heterogeneous. So there are some regions that are very different from other regions, and this contribute. And there are several other factors. The tumor microenvironment is not only made of immune cells, it is made up of stromal cells like fibroblasts, blood vessels, and nerves that contribute to tumor growth. And therefore, if you treat with immunotherapy, you have these stromal cells that are at the background. You are treating with anti-PD-1 and you are thinking that anti-PD-1 will work. But there are several cells in the tumor microenvironment. So if you are a biomedical scientist interested in cancer immunology, you can ask the question, what cells in the tumor microenvironment that promote or enhance resistance to checkpoints? So, for instance, you can look at even platelets. You can look at pericyte. You can look at muscle, even muscle. You can look at whether the present or even fat, adipose tissues. You can look at anything at all and see whether they have implication of immunotherapies. An example of a study you can conduct is you can take patients who are you can take, let's say, melanoma patients which are undergoing immunotherapy or anti-PD-1 treatment. Then you can look for responders, those that will respond and those that will not respond. Now you see whether any of your cell of interest, let's say muscle, fibroblasts, endothelial cells, nerves, or anything are associated with the response to therapy. And this is how discoveries are made. So without wasting much time, I'll end the part one. And a summary of the part one is that immunotherapies are good, but they do not benefit every patient. And the reason, there are several reasons, but one of the reasons that got people attention is that there are nerves in the tumor microenvironment. So I'll go to my part two, which is cancer neuroscience. So this part, I like talking about this part because my whole PhD training has been in this, cancer neuroscience. But of course, cancer neuroscience was born, I would say it was born from cancer immunology to some extent. So cancer neuroscience, what is cancer neuroscience? It's the study of cancer, how cancers interact with the nervous system. And a picture you see here is pancreatic cancer. And what you see here is nerves that are floating, and the other yellow-like or orange color you see at the background is tumor cells. And this beautiful picture was actually taken from one woman from Oregon who published this paper, her paper in JCI Insight, who showed that, cancer cells can grow into nerves, and what they do is they get nutrient from this nerve so that they can grow. And I'll introduce you to cancer neuroscience from my second talk. So, the nervous system promotes tumor growth. So I'll make this a story. So in 1897, a medical doctor called Hu Yong from Johns Hopkins University, he's a pathologist. So he was going through tissue sections. You know, if you have been to pathology lab before, they have slides there. And he says he's a medical doctor. He went through tissue sections, and what he saw was very over. This is over 100 years. Was very crazy. He saw that most of these tissues were highly innervated. There were nerves in these tissues. So he found it very interesting and reported it. Nobody cared about it. So that's how, I'm taking you back to history of how this cancer neuroscience came about. So after Hugh Young actually saw that there is the presence of nerve in tumors, then he was like, what is happening? At that time, technology was low. People didn't know anything about even the immunotherapy and anything. So he just left it there and kept working on his patient. Over 100 years later, in 2001, Ayala Gustavo from the MD Anderson Cancer Center cultured cancer cells with nerves. And what he saw was that the tumor cells prefer to grow around the nerves. And what happened is that these tumor cells you see here actually secrete factors that actually drive the nerves around it. And the nerves also secrete factors that promoted the tumor growth. And some of the factors you see here are neurotropins and other stuff. And what happened is there was a bidirectional relationship between these. After 2001, which is 100 years after Huyon discovered this, multiple papers have been published. And this have actually shown that the presence of nerves promote tumor growth. Indeed, this whole publications, about several of them, you can see from Ayala's paper in 2001, Michelle Monji from Stanford, multiple group Venkatesh Harvard, and multiple groups in MD Anderson, France, Germany, Frank Rinkler, and other big labs have published multiple papers, as you can see at the top, showing that the presence of nerves promote tumor growth. Actually, the field of emergence, so that resulted in the emergence of the field cancer neuroscience in the last 10 years. So it's a pretty new field, which is very exciting. And actually, the field of cancer neuroscience was featured in the Science magazine. Science magazine actually features hot topics and hot discoveries in science. And they titled it The War of Nerves, just highlighting that amazing work have shown that nerves promote tumor growth, and therefore we should fight against nerves, especially in cancer. And this picture you see here in the Science magazine is showing a nerve where tumor cells grow around it. So myself, multiple groups across the US, Europe, and other places, Asia, especially to China, Japan, people are studying how this nerve promotes tumor growth. If we are able to understand it like people did for checkpoint blockage, we can also look at ways to target nerves to reduce tumor growth. So that gave birth to the field of cancer neuroscience. So let's delve deep into it. So what happens? So I'll take you back to, because cancer neuroscience looks at how the nervous system promotes tumor growth, I'll take you back to the nervous system. And the nervous system is actually made of the central nervous system, which is made of the brain spinal cord, and the peripheral nervous system, which can be divided into autonomic and somatic. Somatic is something mostly you can control. An example, what happened is in the somatic, we have the sensory, which is more of the input, and the motor, which controls, the skeletal muscles. But in the peripheral nervous system, we have the autonomic part that we cannot control, and it is branched to the sympathetic nervous system and the parasympathetic nervous system. So overall, the nervous system, at the right side, we see that almost every organ is innervated. They have nerves, and they have both sympathetic and parasympathetic nerves. And therefore, what this means is that if any of these organs you see here indeed gets cancer, the nerves can begin to accumulate, and that can promote tumor growth. And like you may all remember from your anatomy or physiology class, the sympathetic nervous system is the fight or flight, which is the aggressive ones, and the parasympathetic system is the sleep mode, which is the less aggressive one. So evidence from cancer neuroscience have shown that sympathetic nerve activate tumor growth, and they are characterized by the marker. If you want to stain them or if you want to identify them, they express the enzyme tyrosine hydroxylase. So, as a biomedical scientist in Ghana, wanting to understand biomarkers of cancer growth and progression, you can look at these biomarkers like tyrosine hydroxylase, choline acetyltransferase, which is a marker of parasympathetic nerve, and TRPV1, which is a marker of sensory neuron. You can see whether these markers are associated with stage, disease progression, and even survival in multiple cancers. But what I'm trying to say here is that sympathetic nerve has shown to promote tumor growth. If you have more sympathetic nerve, the tumor grows faster. Um, Parasympathetic nerve evidence have shown that it slows tumor growth. So if you have more parasympathetic nerve, the tumor grows kind of slower. Then more sensory nerve, the tumor also grows very fast. So what I'm trying to say here from this slide is that different nerves have different impact on tumors. And sympathetic and sensory nerves promote tumor growth, whilst parasympathetic nerve inhibit tumor growth. So I'll show you some evidence from cancer neuroscience that show that sensory nerves promote tumor growth. So the left side you can see here in green is CGRP, which is another marker for sensory nerve. And these are different types of mouse breast cancer. And even it is same in human. And what happened is you can see these green guys, they are big nerves. These are chunk of nerves. You can even see here, it's great. This is human breast cancer. You can see a huge chunk of nerves that are accumulated in the tumors. And what they are doing here is, for instance, you can see here where you see the tumor is blue and then the nerve is green, and you can see that they are just growing around it. So the nerves are promoting tumor growth. This is breast cancer. So what one group in Columbia University actually did, which is Pan Randaman, who was a postdoc in Tavoyo's lab at Columbia, what she did was that she actually used a chemical called capsaicin, and capsaicin can inhibit or desensitize sensory nerves. And what she saw was that the tumors were very low. You see, the size of the tumor at any time point is smaller compared to when you treat it with a control, which we call a vehicle, basically PBS or water. So what happened is, if you are blocking sensory nerve, the tumor slows down. And what happened is, even metastasis to the lung is also reduced when you inhibit the activity of sensory nerve. So this evidence showed that indeed sensory nerve, the presence promotes tumor growth, and if you block it or inhibit it in any form, you reduce tumor growth. So another thing is that the sensory nerve, not only that their presence reduces tumor growth, but the sensory nerve can produce peptides. An example of peptide produced by sensory nerve is CGRP, which is called calcitonin gene-related peptide, and substance P. And what happened is, what they do is that they promote tumor growth. And what happened is, if a mouse, this study was done in mouse, where they took mice that do not have CGRP, which is TAC1 is a gene that encodes for CGRP. So they took a mouse, knocked down CGRP, and what happened is there was a smaller tumor growth and metastasis to the lung was significantly reduced. Similarly, substance P is also produced by sensory nerves. And what happened is, in an intact mouse, if you treat it with a control or anti-substance P, even though there is a presence of sensory nerve, if you block the release of substance P, there is a smaller tumor growth and metastasis to the lung is also significantly reduced. So this evidence, my talk is showing data from the field that indeed shows that, which, like I said, these are none of my own data. I've done some of these experiments, but like I said, two reasons. It is not published yet. And since it's not published yet, it doesn't belong to me. It still belongs to my professor and the lab, and they have to authorize it before I can present it anywhere. But this is amazing data showing that the presence of sensory nerve or their peptides promote tumor growth. And if you block the peptides, like if you inhibit substance P or calcitonin gene-related peptides, CGRP, which are all peptides produced by sensory nerve, you inhibit cancer growth and metastasis to the lung. Now let's shift gears to the sympathetic nerves and let's see some evidence. So first thing is they activate one group. This woman she's called Erica Sloan from Monash University in Australia. They use a stress. They activated the sympathetic nervous system. And if you remember your pharmacology, there is a drug called isoprosterol, which is an agonist of the sympathetic nervous system. What it does is it binds to beta adrenergic receptors, and what it does is it activates the sympathetic nervous system. And what happens is if you treat them with isoprosterol, you see that the tumor size, which is luciferase here, is very high, and metastasis to the lung is very much increased compared to the control. So basically, another experiment they did is first, this is using the chemical or sympathetic agonist or adrenergic agonist. Another experiment they did was to stress the mouse. And how they did this is they put it in a cage, they will shake the mouse so that it is stressed. When you are stressed, your sympathetic nervous system is activated. And what happened is the stressed mice have very big tumor compared to the control mice, mice that had normal life. And what happened is in stressed mice, the tumor was very big, metastasis to the lung and other tissues were very significantly high compared to the mice that were not stressed. So the evidence shows here that sympathetic nerve activation via stress or even adrenergic agonist promote tumor growth. So what is the mechanism? I've been showing you multiple data sets showing that nerves promote tumor. What happens at the background at the molecular level? So multiple things happen. The nerve enhanced proliferation. They activate downstream signaling in cancer like PI3K signaling. They can form connections with the tumor cells, especially in brain tumor, where they can release electrical signals to promote tumor growth and metastasis. They can enhance epithelial to mesenchymal transition. They can inhibit apoptosis of cancer cells so that they don't die. And most importantly, they can enhance immunosuppression and T cell exhaustion. So in the right hand here is the top side is a tumor cell where there is no nerves. There are no nerve bundles. And the bottom side is we have accumulated nerve in blue. So the beta 3 tubulin is a marker for nerve. And Ki67 is a marker for proliferation. So proliferation means if cells are abnormally growing and dividing, they express proliferation markers like Ki67. So what these people did was that they took patients with nerves and took the tumor regions that are nerves and regions that do not have nerves. And whenever there is nerves, you see that the cells are proliferating more. They have the red marker, which is Ki67, which is a marker of proliferation. So you can equally do this, and anything, most of the stuff I'm showing here was conducted in the Western world. So as a biomedical science, and if you quantify, you see that the presence of nerves, nerve bundles, increase proliferation. And if you're in Ghana, you want to do your master's or anything, you can think about how nerves enhances any of these pathways in cancer. The last part of my talk, which is very simple, but more important, is what is the impact of nerves on immunotherapy? Remember, I've already spoken to you about cancer immunotherapy, which is the first part of my talk. How does this cancer neuroscience interact with the immunotherapies? So what is the translation? First, I'll talk to you about perineural invasion, which is very important for the people in biomedical sciences and biomarker discovery. I'll talk to you about how beta blockers enhance resistance to immunotherapy, and then talk about how some nerves enhance resistance to checkpoint. So first thing I'll talk about to you about the translation aspect is the term we call perineural invasion. So perineural invasion is a situation whereby nerves grow into tumors. So you can see it's called perineural invasion, PNI. And a picture here in H&E or even in immunohistochemistry show that S100B is a marker for nerve 2. And you can see that this nerve is growing around the tumor cell. So basically the nerve is invading the tumor. So they took patient Nisha De Silva, who is a pathologist and a professor here in the United States. What she did was they took tumors from patient, then identified with the assistance of a pathologist. They were able to identify tumors that had perineural invasion. And what they did, which is the accumulation of nerves around tumors. And they were able to then also get patients that had less perineural invasion or even no perineural invasion at all. Then they compare the survival. And you see that patient with perineural invasion, which is red, they died faster compared to patient without perineural invasion. What this finding is telling us is that the presence of nerves in promotes, the tumor growth and it reduces the survival. Now, as a biomedical scientist in Ghana, one question I want you to think about if you are thinking about your master's or PhD, there are multiple cancer data patient tumors sitting at comfort. They have slides. Patient came for diagnosis. They probably did surgery or resection and took tumor cells, stained them on a slide, H&E, or even they have slides sitting there. If you are planning of doing your master's, PhD, you can take perineural invasion and see whether it could be a diagnostic biomarker for specific cancers, whether it can be associated with patient stage of the disease because Ghana patients are known like breast cancer, most patients present with the advanced stage of the disease. You can compare it to the early stage of disease and see whether perineural invasion can distinguish between different stages of disease and whether it's even associated with the survivor. And even if patients are undergoing checkpoint therapy, you can ask the question, does the present before they take anti-PD-1, you can actually ask the question, you can screen for perineural invasion. Now, some patients will have perineural invasion, others would not. Then you give them the anti-PD-1 therapy and see at the end which of them will respond better to the therapy. So that's the first translation I want to talk to you about, which is the biomarker. So I want you all, if you're interested in the field of cancer neuroscience, think about how perineural invasion can be used as a diagnostic biomarker that can enhance patient stratification for therapy, that can be used to monitor whether patients are even responding to checkpoint therapies and even different therapies in multiple cancers, especially among African population where these studies are not being conducted at all. Then another thing is nerves can enhance T cell exhaustion. Sorry that I put M here, but it's exhausting. I'm sorry for the typo. But this is one study from Susan Keck's lab at Scripps Research in California. And what they did was that they took patients with cancer, and what they did was they stained for tyrosine hydroxylase, which is a marker for nerves, sympathetic nerves, and then stained for CD8 T cells, and also added markers of exhaustion. And what they show was that in patients that have high sensory sympathetic nerves, the CD8 T cells were more exhausted compared to patients that had low sympathetic nerves. So basically, this is telling us that the presence of sympathetic nerves enhance CD8 T cell exhaustion. That this paper was published in 2023. Another paper published in 2021 by Sebastian Talbot's lab in Canada actually have shown that CD8 T cells expressing receptor RAMP1, which can respond to sensory neurons, they actually have big tumors. And if you knock out or block RAMP1 CD8 T cells, you reduce tumor growth. And even this was conducted in mice, but they also took human patients and patients with high RAMP1, they die faster compared to patients with low RAMP1. All because if your RAMP1, if you express RAMP1, the CD8 T cells are able to respond to the nerves and they get more exhausted compared to the other CD8 T cells that do not express RAMP1. So this evidence, both from sensory and sympathetic angle, shows that nerves can enhance T cell exhaustion. One last part is a clinical trial by Erica Sloan's group from both Australia and Columbia universities. And what they show was that they took breast cancer patients. The one problem with breast cancer is that the gold standard for treating breast cancer, especially advanced breast cancer, is resection, where they will just do surgery and then take off the tumor. But one challenge is that there is reoccurrence. After you take the primary tumor, the tumor comes back again. That's what we call recurrence. So knowing that the sympathetic nerve activity block enhance tumor growth, Erica Sloan's group did a very smart study where they grouped patients into two: breast cancer patients that received beta blocker and those that did not receive beta blocker. And more important, I want you to see here is CD8 T cells. Upon beta blockage, the patient had more CD8 T cells. They have less recurrence, tumor recurrence. So what is telling us here is that the beta blocker inhibited sympathetic activity because the beta blocker, they use propranolol. And propranolol will inhibit sympathetic nerve activity. And what happened is that the CD8 T cells are actually increased. And what they also found was that the patient had less tumor recurrence compared to the patient who were not on beta blockers. The last point data I want to tell you is that in the first part of my section, I told you that anti-PD-1 therapy works, but not all the patients benefit. Why? Because of nerves. So Barunch, which is a postdoc from the Moran Amit's lab at the MD Anderson Cancer Center, last year published a very big paper in Nature, which they show that sensory nerve can actually enhance what do you call it? Tumor growth. And actually, if you actually denervate a mouse, so if you cut the nerves, then add anti-PD-1 therapy, you see that, which is denervation, which is the second group, the tumors grow very slow. But if the nerve is intact, and then if you have a nerve intact and without treatment, the tumor is very big. What this whole finding was telling us that if you treat with anti-PD-1 treatment, in the presence of nerve, the tumor grows. But in the absence of nerve, or if you block nerve activity, the tumor grows slower. In the same study, they showed that this sensory nerve can actually induce injury and IL-6 signaling. And if you block anti-PD-1 with an IL-6 inhibitor, receptor inhibitor, you enhance response to the checkpoint therapy. So this is telling us that just by treating with anti-PD-1, it may not work because sensory nerves can enhance the resistance to anti-PD-1. But if you get anything that can block the sensory nerve, we can enhance the response. So a summary and avenue for cancer neuroscience in Africa, since I'm talking to a group where most of you are from Africa, is that I think I've been able to tell you that indeed nerves promote tumor growth and metastasis. Therefore, are nerves a new hallmark or is cancer neuroscience and the presence of nerves a new hallmark of cancer? Because I already showed you the 14th hallmark. The answer today is no, but very soon I'm sure that nerves will be a new hallmark. One question I want you all to start thinking is, what is the diagnostic potential of nerves and perineural invasion, especially in African breast cancer patients? Can perineural invasion be used as a diagnostic biomarker for monitoring treatment response, disease progression in multiple cancers? Then, is there association between nerves and clinical disease progression in African population. So, for instance, you can take breast cancer subtypes like triple negative HER2, ER, and PR positive. And also even take breast cancer stages, stage 1, stage 2, stage 3, stage 4, compared to healthy controls. Is there a difference in the tumor innervation and even perineural invasion? These are all studies you guys can think about, and they are cheap to do because you just need to stain for these nerves. And given that there is the Ghana National Research Fund, which the current president of Ghana actually established a committee which they funded $100 million to support research across different fields in Ghana, which I know for sure that majority of this money is going to be channeled to health and biomedical science research. These are ideas you guys can think about and then move on to advance the field of cancer neuroscience. So without wasting much time, I would like to once again thank the ACRI for inviting me, and I'll thank the participant for attending to listen to my talk, and I'm happy to take questions. Thank you.
Thank you very much, Stephen, for this wonderful presentation. There's a lot, and I believe that we've all learned something new. Please, the floor is open for questions. Um, uh, if you also want to type in your question in the chat box, you can do so, and we'll read it out. So please, the floor is open for questions. But well, let me ask my question first. So, Stephen, please, what is the risk of the denervation? I heard you mention that some of the the research work that they've been doing, they were denervating all of it and then using the Kaplan-Meier curve, we were showing some positive outcomes. But then denervating, then won't it also affect the normal functioning of the person? And the second question is, I saw most of the research is being focused on the sensory nerves. But what about the parasympathetic? Since they are able to limit tumor growth, are there researchers that are targeting those ones to kind of limit the tumor growth? Thank you.
Yeah, thank you. Oh, well, that's a very great question. So I think these are three questions. One, the innovation. So whenever I travel, one big challenge in cancer neuroscience is translating it, right? You see, for instance, people say sensory nerves promote breast cancer growth. So do you want them to denervate breast? No, because you paralyze the patient. So those denervation experiments were done in mice, but some of the blockage have been done in humans. But it's not translational and even the beta blockers. For instance, the breast cancer patient in that clinical trial where they actually use beta blockers to inhibit the how do I put it? The inhibit the sympathetic nerve activity. What happened is that beta blockers actually reduces the blood pressure. So some of the breast cancer patient, if they didn't have hypertension, now their blood pressure was very much lower. So the challenge in this field of cancer neuroscience is side effect. So oh well, your question now, how do we address this? So that brings to my project. So what I'm doing now is that's where wherever I travel with my research, it's very novel and people like it, is that instead of targeting the nerve themselves, because you can paralyze the patient. Trust me, if you denervate human being, they are going to paralyze. If you touch the breast, they will not feel anything. So what happened to, how do I put it? What we are doing is that we have actually found that some immune cells and even endothelial cells rather are the cells that they produce neurotropic factors to call the nerves into the tumor. So instead of targeting the nerves directly, we rather target the factors that drive the nerves. So I don't want to go detailed into that because it's my whole work is very novel, but instead of targeting the nerve directly, we rather target substances that drive the nerve so that we don't even get the nerves into the tumor directly. So example is that endothelial cells can produce neurotropic factors to drive nerve into tumor. So instead of denervating the person or treating with beta blocker that has systemic effect, you can rather treat it with anti-VEGF, which is vascular endothelial growth factor, so that we can block it. My project is also looking at specific immune cells that produce neurotropic factors to drive nerve into the tumor. So now, instead of denervating the person, I can just, I'm proposing to actually use a neurotropic inhibitor. Then we don't get the nerves in the first place, so that we don't even denervate. And I think the other question is that the parasympathetic, yes, is very, very interesting that the parasympathetic um, inhibit tumor growth. There is some evidence which I didn't put too much here, but why most people don't look at it is that it hasn't really given much consistent results across tumors. So unlike sensory nerve, which if you activate sensory in breast, renal, kidney, brain, anywhere, it promotes tumor growth. Sympathetic, and also the sympathetic to the same, both of them promote tumor growth. In the parasympathetic, in some cancers, it inhibits tumor growth. In some cancers, it promotes tumor growth. So it's very dicey. And that's why if anybody here is interested in a master's or PhD, get tumor cells from Confanoche or anything. Of course, you guys cannot do the mouse work because Ghana, we do not have that resources. But see patient with very big tumor and very advanced stage. Do they have more sensory, sympathetic, parasympathetic? And you can help answer. So I don't have a question for your third one, which is the parasympathetic, because there we still need, like I said, cancer neuroscience is still new. So we still need more evidence to show that whether parasympathetic, whether it promotes or inhibits. The evidence so far is a mixed, 50-50. But for sensory and sympathetic, they promote tumor growth. And like I said, your first question is very excellent. We don't want to denervate this patient. We don't even want to use the beta blocker because if the patient blood pressure is normal, now the blood pressure will go down. So rather, my approach to address cancer in neuroscience is that I am identifying immune cells that can call the nerve into the tumor so that we can target these neurotropic factors produced by the immune cells so that we don't even cause, systemic effect. So I hope this answers your question. Yes.
Very much. Thank you very much. That really answered the question. And we have one question from Ernest. It's in the chat box, so I read. It says, in standard histopathology report at tertiary centers like Kolebu, perineural invasion may be simply noted as a prognostic marker. Based on this new paradigm where nerves actively feed the tumor, should we be demanding more detailed reporting on nerve density and PNI from our pathologists to better stratify patient risk? Thank you.
Yeah, so that's a very good question. Yeah, so your pathologist first must know that there is even a field called cancer neuroscience. So last year, the American and Canadian Association for Pathology actually met at a very big conference in, I think it was in Alberta in Canada, and it comprised of all the big pathologists across America and Canada. And what they did was they actually sat down to define the standard definition of perineural invasion. And they did what we call a plus system. So you can take the patient tumor, score it as +1, +2, +3, +4. So now the big question is, are the pathologists in Ghana first, do they even know that something like this exists? So yes, you can request them to do, but they must first be trained and must be aware. And that's why research should be collaboration. So here, research is conducted among scientists who are in the bench. They don't care, not that they don't care about patient, but they have no access to patient. Then in collaboration with the pathologists, surgeons, then they come together to identify something new. But unfortunately for Ghana, yes, that's why. So I feel like for now in Ghana, I don't think any pathologist or a surgeon will be able to do this because they don't even know, to the best of my knowledge. But if any of you or any of us happen to conduct a very big study in Ghana and show that, yes, perineural invasion is associated with worse prognosis or is associated with poor and or early advancement or even survival, then that paper can be published. then pathologists in Ghana and across Africa can now meet to also define their criteria. So I feel like Africa and Ghana, we are a half step behind of discovery. And today, yes, if you request it, yes, you can say pathologist. Can you look for perineal invasion? The question is, do they even know it at all? So I feel like it will take a little while, especially when it is standardized internationally. They can adopt it. Yes. So I hope this answers the person's question.
All right. Thank you very much. So please, the floor is still open for questions. If you have questions,
Yeah, feel free to ask any question in cancer neuroscience. Like this is a super summary, and I'm sorry, I know that it's a very high level. I didn't take it to the beginner level, but I really wanted you guys to. This is not a class, it's not a cancer immunology, cancer neuroscience class. So I didn't take it from basics, and I also didn't make it like a conference presentation, but this was to just give you some insight about something that is being done here and tell you what you can do, especially if you are thinking about doing your master's, if you are thinking about doing your PhD, if you are even an independent researcher or a pathologist or a biomedical scientist, a nurse or anything, things you can look about, you can just get ethical approval and start. And most of these tissues are sitting in Konfanochi and Kolebu. You can just get them, stain them together with a pathologist who might be even be interested in this. Yes. I think somebody raised their hand.
okay, please, if you raise your hand, please ask your question. If you raise your hand, please ask your question. Perhaps I missed it. Um, please, if you raise your hand, kindly ask your question. I hope the network has not affected anyone's. Okay, yeah, Belinda, please ask your question.
Okay, thank you. Good evening, everyone. And thank you to our presenter, Mr. Stephen Opoku. Please, my question is to those of us that are interested more with hemato-oncologists, especially with the leukemias, which are not tumors. Does that mean that the field of neuroscience cannot be explored in terms of that because you don't have nerves invading or invading the blood, right? So I want to know how somebody who is interested in non-tumor cancers can explore this field. Thank you.
That's a very great question, Belinda. Yeah, that's a very great question. You are talking about, of course, what I spoke today is solid tumors, but you are talking about leukemias, which are more liquid tumors and fluent. And what happened is that the origin of leukemias is from the bone marrow, most of them, because it's abnormal white cell development that mostly lead to these leukemias. And you can't believe me that bone marrow have huge amount of nerves. Whether the nerves in the bone marrow influence leukemia progression and metastasis to different organs, or even it reprogram the leukemic cells, I don't know. Next, there are some leukemias like um, Hodgkin's lymphoma, which happens in the lymph node where you have Stenberg cells, right? And remember, these lymph nodes are actually already innervated, and whether the presence of nerve influence that nobody knows. But of course, unlike the solid tumors, like I know for sure that, of course, leukemic cells float around the body in the blood. So within the blood, they cannot, perineal invasion may not be a biomarker. But remember, leukemic cells actually accumulate in liver that can cause anemia and even this bilirubin, we call it jaundice, right? So whether the presence of nerves at different organs where these leukemic cells evade play a role, we don't know. So it's a good research you can look at. Probably take bone marrow tap from this leukemic patient and see whether patient with that worst disease, patient who die faster, the bone marrow is actually enriched with nerve. That's why those patients, the leukemic cells actually proliferate faster or anything. Nobody knows that. Yeah. But a great answer for you is that for sure, to the best of knowledge, nobody has studied the role of nerves in leukemia. But you can study it, of course, not in the blood because yes, there are no nerves in the blood. But remember, the leukemic cells evade both the bone marrow, different organs. So we can still look at in these organs and see whether it is associated with patient characteristics. Belinda, I hope this answers your question.
Yes, thank you.
For sure.
Okay, so please, any more questions? Any more questions? I think we should ask questions. So, all right. So, Stephen, my next question, this is a bit far-fetched, but is there any linkage between cancer neuroscience and cancer genetics? Especially as cancer neuroscience has more to do with cancer immunology, but does it stretch to cancer genetics, the basic at the molecular and basic level? Let me put it that way. Thank
For
you.
sure. It's something I didn't want to go to because as soon as you start going into genetics, there are huge pathways you need to talk about, right? Which I didn't get there. But it has a very huge role. When the nerves, I didn't get there, but probably you saw a pathway I showed that PI3K signaling, where you have several transcription factors and molecular switches that can be activated, like the JAK-STAT pathway, PI3K signaling, AKT. These nerves so for instance, sensory nerve can produce CGRP. CGRP can bind to RAMP1 on the cancer cell. It actually causes several genetic and molecular changes within that cancer cell, which is one angle people look at. So the neuroscience of cancer has implications across almost everything you can think about in cancer. Genetics, cellular level, molecular level, cellular level, anything you can think about. It has metabolic even implications. Some studies have shown that nerves can actually dysregulate glycolysis in tumor cells to cause more metastasis. So it has some studies have shown that sympathetic nerves can actually dysregulate fatty acid metabolism. And the implication is that the downstream tumor cells dysregulate TCA cycle, and the end result is that they actually evade the immune system and enhance their metastasis. So molecular level, there are a lot of evidence there. Even another very exciting thing is bioenergetics. So cancer neuroscience, it has implication of bioenergetics, and bioenergetics mostly study the mitochondria and energy transfer, okay? And one big study, which you guys can look it up, is the nerves can release mitochondria. And you know, mitochondria have energy. It's the powerhouse of a cell, right? So when tumor cells are stressed and weak enough, the nerves can actually release transfer mitochondria cells get energy and they can grow and metastasize. So we call it the interaction between cancer neuroscience and bioenergetics. So yeah, there is a huge relationship between, cancer neuroscience, genetics, bioenergetics, metabolism, a whole lot. Yeah.
Wow, that's great.
Yeah.
When you when yeah.
Sorry to cut you in. So that's why I love this field, because no matter who you are, if you are a nurse, so there is one professor at UAB Nursing which I went to the International Cancer Neuroscience Symposium, which was people from 42 different countries. And what she's doing is that the chemotherapy we are talking about and immunotherapy, sometimes they can induce pain, okay? So she, as a nurse managing cancer patient, she's looking at how these nerves, biomarkers of pain in cancer, because some of these cancers, like pancreatic cancer, a symptom of this pancreatic cancer is pain. And the pain is because of the nerves that are infiltrating there. And she is a nurse, so she's looking at bio. So, pain is always asked by a score, a questionnaire, okay? So the patient come and you ask them, you touch their pancreas, the area, the abdominal area, and ask, do you feel pain? Do you feel this? they think the questionnaires are not accurate because somebody's pain level 10 is relative to somebody's pain level 4. So what she is doing is she is collaborating with biomedical scientists to look at markers, actual markers, such as neurofilament L, and see how those biomarkers can predict pain better. So it's super interesting that no matter who you are, if your background is pure biology, nursing, pharmacy, anything, you can link cancer neuroscience to your field. So when I came to cancer neuroscience, most people were not really looking at the immune system. And therefore, as immunologist, I just look at how the immune system promote nerves. Simple. So it's something no matter your background, you can directly fit in, and it's very interesting, and that's why it's booming in the last 10, 15, 20 years. Yes.
Thank you very much, Stephen. So please, the floor is still open for questions. If you have any question, please, you can ask. Stephen is ready to answer them. We have some few minutes more and then. Okay, yes. So Reginald, please kindly ask your question.
Reginald, I don't think we can hear you.
Can you hear me now?
Yes, we can.
Yeah, so thank you very much for such an insightful presentation. My question is, since now we know neural networks or nerves do actually feed tumor markers, can we also look at the other side, whereby we look at neural activity to predict the onset of cancer or the recurrence of cancer or other tumor markers? Could that be possible?
Yeah, that's Reggie, that's an amazing question. So some groups are doing that, but not too much, where they just reprogram. Some people will take mouse, okay? One guy, postdoc in our lab, she would actually activate the nerves, take the mouse. There are ways to do that. We have a term called chemogenetic activation, where you expose light to specific part of the brain. And what you will find is that if you overactivate it, and cancer is not caused by a single thing, right? It's a compounding mixture of factors. And upon doing that, those minds that are actually activated where the nervous system is overactivated, those minds get tumors very fast. So what happened is, yes, for sure, activating the nervous system definitely can activate and change tumor markers. But whether that associates with survival and all those things, if you are interested in it, yeah, you can look at it. Does that answer your question?
So he has replied, thanks. Yeah, so I think that answers this.
Yeah, and one very funny thing in cancer neuroscience, which I didn't talk about, but I want you guys, if you didn't hear anything, go and read about this. So there is brain tumor, okay? I want us to discuss on this. Brain cancer or brain tumor is a tumor that happens in the brain, and the brain is made up of nerves. We all believe that the brain has huge millions of nerves, true or false? It's just a discussion, so I want somebody to respond.
Yeah, true.
Yeah, that's true.
Now, based on our discussion, we know that neuronal activity increase tumor growth, true or false?
True.
So the more you think, if you have brain cancer, the more you think, the more you are likely to die, true or false?
That is true.
Do you guys understand? So there is an article. So you guys go and just search about this. The more you think, the more you are likely to die from brain cancer, because you are activating your nerves in the brain. Because if you have brain cancer, the cancer is sitting in the brain, and they've shown that nerve activity in the brain promotes tumor growth, right? So now, if you have brain tumor and you are using the same brain to overthink, so if you have brain cancer and you have broken heart, you are likely going to die early. Is this very interesting? So when you go, you just go and Google and read about that. The more you think, the more you are likely to die from brain cancer. There is a whole journal, it's a news article that covered that. Yeah. And that was championed by Frank Winkler from Germany, and Michelle Mongi from Stanford University. So it's so exciting, you know.
All right,
Yeah.
not to cut you, Stephen, but Belinda has another question. She says, as someone interested in cancer neuroscience, where do you advise they start from? I'm trying to know more about the field.
Yeah, so good. That's very nice. So it's very unfortunate that this my meeting happened now. So there is a very big conference on cancer neuroscience called the International Cancer Neuroscience Symposium at the MD Anderson Cancer Center in Houston. It's the biggest cancer center in the world. That's where I went to present my work in February. And it happens every 2 years. So if you really want to start, I would recommend that you read about International Cancer Neuroscience Symposium 2026. Go to the website, and when you go to the website, you see just play about the website. You see professors who came to present, you see people presented, and then you can start looking for their works. And I mean, I'm very excited and happy to mentor anybody who is interested in the field. Anybody who wants to, if you are really, really interested in cancer neuroscience, and let's say you don't know where to start, or if you are interested in doing something on cancer neuroscience, because all this staining, the nerves, the activation of the nerve, the chemicals we use to activate them, you need a little bit of training to know how to do it back in Ghana, right? Trust me, it's the cheapest thing you can do. If you are doing a master's in, let's say, hematology, oh, well, how much did you buy your reagents while doing your master's? Probably he did. Oh, if you would not spend less than 10 to 15 or
Yes,
20,000.
I mean one reagent will cost like 8,000 cedis. That's one reagent, yes.
And how many did you buy?
Yeah, buy like two. That is for only one marker. So the other marker, depending on your sample size, of course. So
This
yes.
one, you can even do just simple H&E and IHC, very cheap. So if just to go back to Belinda's question, if you're interested in cancer neuroscience, of course, first you just read about paper nerve promotes tumor growth. Just start searching nerve promote breast cancer growth, promote brain tumor growth, nerve promotes renal cancer. You will see a lot of papers there you can read. That's one. Two, just feel free to, I can send you all my email on in the chat, 07 yeah, 275 at gmail.com. And I'm more than happy to send you more than 1 million resources for you to read. And if you are interested in, studying cancer neuroscience as a PhD or master's, you must definitely show that maybe you know someone because trust me, all the big people in cancer neuroscience knows me. Almost everyone in cancer neuroscience. Everyone in cancer neuroscience knows me, especially the big people in cancer neuroscience. So if you are like, oh, Stephen is your mentor, let's say you are applying and there are specific schools that are really much, they have a lot of, like MD Anderson Cancer Center. If you apply to their biomedical science program, and let's say you were able to just do breast cancer stage 1, 2, 3, 4 in Ghana population, and you show that maybe you did your master's, then show that, oh, stage 4 is highly innervated with sympathetic and sensory nerve compared to parasympathetic and all that. Simple analysis. Trust me, you are likely going to get a PhD compared to anybody who has spent 20 years doing anything unrelated to cancer neuroscience and then wake up any day wanting to do it. So, but anybody interested in, you can start reading, but if you need questions, yeah, for sure, feel free to email me and I'll send you more than enough resources.
Fantastic.
Thank you.
Yeah. Yeah, I mean, the most interesting part why I agree to talk about this is not about the presentation or whatever, because some of the things are hard to understand. But, to share and all of us have a discussion about the field, people's interests, people who want to do cancer neuroscience. And the beautiful thing is, irrespective of your background, because one challenge in cancer is that when patients go for surgery, immunotherapy, chemotherapy, there is pain. And cancer-induced pain, if you're a nurse, you may want to study that even during your undergrad thesis to see whether a pathologist may want to give you all this perineura. You can collaborate with students or pathologists or any other people. Then you can look at whether the presence of specific nerve, let's say a patient with perineura invasion, what is the pain score in those patients compared to patients with no perineura invasion? So it's so exciting that anybody at all can fit into cancer neuroscience because it's so new in cancer.
It's Stephen, we thank you very much. We are grateful. You've really done a fantastic job. God richly bless you. Please, in the absence of, are there any questions? Let me ask that before we can allow Stephen to go so that he can go and watch the Ghana match, if only he will go and watch.
Oh yeah, for sure. I'll definitely go and watch Ghana match for sure.
Okay.
I hope my email address is correct. Oh, it's Steven275. Yeah, for sure it's correct.
Yes. So please, Stephen has placed this email, how do you call it? Email account in the chat box. Kindly copy and then save and then reach out to him. Trust me, he's a fantastic guy. When you reach out to him, he will reply you fast. Yeah, so that one day is rest assured. And I'm sure you can have a good discussion on the cancer neuroscience, the prospect, and he'll guide you on how to, either join him there or kind of start a research here where you can collaborate, right? And then
Yeah, I mean, yeah, for sure. I'm always open to collaborations. And if people are interested in graduate school because you want to do a master's, everybody is doing the same old school, the 1991 masters. You still your grandfather did masters in a specific program. You also want to do the same route and do the same research. You can do your masters in hematology, molecular biology, immunology, biology, anything, nursing, but can you integrate cancer neuroscience in whatever field you go? With that, if you finish, I promise you, if you want a PhD, it's going to be an easy catch because you are going to have an SOP for the entire of Africa. Maybe nobody else has that kind of SOP. You are going to present, when they see your SOP, they'll be like, oh, wow, this guy really wants to do something new. Then no, you will not have competitors. Yeah. And I think if you don't remember any biomarker from here, just remember perineural invasion and how to quantify that. Yes.
right, he and I. Please, any question? Any question? Or yes, any question for Stephen before we let him go? Okay, Stephen, I think we've exhausted our questions. Once again, I want to say thank you very much for making the time. I'm sure another time when we reach out to you, we're happy to. Yeah.
Oh, for sure. I mean, I'm always hopefully, yeah, I'm always available. So for sure, I'm more than happy to, share this. Yeah. And I'm sure that by that time my paper will be published and then I can share data from my own things I did with my hand, so which is a very new and different, very new, interesting direction. Yeah.
Fantastic. Okay, so please, in the absence of any question, we say a big thank you to Stephen for doing us the honor to present to us on cancer immunology and cancer neuroscience, mechanistic insights into the diagnostic and therapeutics I believe we've learned a lot. Our next session will be on the 11th. I hope you can also make it. We are looking at AI and nanomedicine in precision oncology. I'm sure you also learn something from that. It's also 5 p.m. GMT, 1 p.m. EDT. Kindly make the time to join us. So, Stephen, once again, God bless you. And I want to say bye-bye to everyone.
All right.
Have a blessed evening. God bless you all. Bye-bye.
All right, bye. All right, bye-bye. All right, see you guys
Bye.
all. Yeah,
Bye.
bye. Happy to
Bye.
talk to you all. Yeah.
Bye.